Welcome back to the model portion of this week's topic, the brain. We will be using some of the models that we normally would look at if we were on campus at CNM. Most of the brain structures can be shown with this particular model of the brain. I do have a couple of other ones that I will show you as we go through the list of terms. I'm going to try to stick to the list of terms, the sequence of terms in the list of terms to make it easier for you to follow. So we begin by looking at the cerebrum, which is most of what you're seeing here. And as we noted, the cerebrum has lots of folds in it and depressions which increase the surface area so that we maximize the amount of gray matter which is found in the so-called cortex of the brain, which is the layer of gray matter just at the surface. Some of these depressions are very large, and these are known as fissures. We have two main fissures that we want to be aware of. The longitudinal fissure is this big one right here that cuts the brain or the cerebrum into right and left hemispheres. The other major fissure is down at the bottom between the cerebrum and the cerebellum. And this is another main fissure, and it is known as the transverse fissure. All the other depressions, little grooves that you see in the surface of the brain, are called sulci, individually known as the sulcus. And so that's these little indentations or grooves. Each one is called a sulcus. There are a couple of particular sulci that we want you to be aware of. This one right here is fairly obvious, kind of halfway back through the brain. That's the central sulcus. And then this one here that separates this lobe from the lobes above, this one is known as the lateral sulcus. So we have a central sulcus about halfway back through the cerebrum and then a lateral sulcus here on the side. The lumps of tissue between sulci, the lumps are called gyri. Each one is referred to as a gyrus. And they do have names, but we're only going to ask you to learn the names of two of the gyri. Again, the ones on either side of the central sulcus. So this one in front of the central sulcus, this gyrus is referred to as the precentral gyrus. The one behind is known as the postcentral gyrus. The cerebrum is broken up into identifiable lobes, and the sulci can help us identify these lobes. So in front of the central sulcus here, all of this is known as the frontal lobe, and the names of the cranial bones of the skull also help you remember these because they're named the same. So frontal lobe here. Behind the central sulcus would be the parietal lobe. Off to the side down here, underneath the lateral sulcus, would be the temporal lobe. And then on the back, way back down here, would be the occipital lobe of the cerebrum. There is a fifth lobe called the insula, but that's deep underneath there, and you have to pull back the temporal lobe to see it, and we can't do that on this static model. So again, the cerebrum is this largest portion of the brain, everything we've been talking about, and as I mentioned earlier, the cerebral cortex is the gray matter that lies underneath or lies at the surface of the brain. And again, this brain is not sectioned, so you cannot see the cerebral cortex, but it would be the gray matter at the surface of the brain. What we can see here is the corpus callosum. Corpus callosum is this comma-shaped band of white matter. And again, this consists of tracts of myelinated axons that are helping the left and right cerebral hemisphere to communicate with one another. We also see a structure called the fornix. The fornix is this curve-shaped structure right here. Let's see if we can zoom in a little bit. So the fornix is this curve-shaped structure right here. I did not talk about this in the video, but the fornix is part of the limbic system, and it helps communicate between the limbic system and the thalamus. Between the corpus callosum and the fornix is another structure I did not talk about, and that's a sheet-like sheet of tissue right here known as the septum pellucidum. The septum pellucidum is actually a wall that sits between the lateral ventricles. Remember, the ventricles are hollow spaces in the brain, and we'll take a look at those in just a second. And the lateral ventricles are the first two ventricles located in each cerebral hemisphere. But right here, they come very close to each other, and there is a thin membrane, essentially, that separates them, and that's called the septum pellucidum. Septum meaning wall, pellucidum meaning translucent. Lucid, luz is a word for light, and so if you were to magically shine a flashlight in the other side of this membrane, you could see the light coming through. It's translucent. So septum pellucidum. As I mentioned, the lateral ventricles exist in each of the cerebral hemispheres, and they are fairly unusually shaped structures. And I have a model of the ventricles for us to look at, and essentially what it is it is a mold of the ventricles. It looks like this, and it's probably the best way to visualize the ventricles. So in this particular model, what you want to do is take away all the solid material that makes up the brain, and what's actually left as solid is what the hollow spaces would look like inside. So imagine if you could fill up all the ventricles with plastic or some kind of material, and then take away all the solid brain matter. This is what would be left. And I just want to point out how oddly shaped the lateral ventricles are. In the video, I described them as C-shaped, and I guess they are approximately C-shaped. They have a little projection on the back side here, but that's what the ventricles look like. At least the lateral ventricles look like inside the cerebrum. And I'll come back to this model several times to highlight the other ventricles as we go. So we'll just leave it back there for now and bring this back forward. All right, so basal nuclei, as I described in the PowerPoint video, are islands of gray matter located in the white matter. We don't really have any good models of basal nuclei, so you don't have to worry about identifying basal nuclei in any of the models. All right, then we proceed to the diencephalon. You want to recall that the diencephalon is part of the forebrain and the upper part of the brainstem, and essentially the diencephalon is this region right here, all of this region right in this area. This is all diencephalon. And the diencephalon consists of several structures, but one thing to remember before you start talking about the solid structures is that at the very center, at the very heart of the diencephalon, is another hollow chamber, the third ventricle. So you have your first two ventricles in the cerebral hemispheres, and then the third ventricle is right here, a hollow space right here in the center of the diencephalon. On our ventricle model, if I superimpose it here, our ventricle model, this area here would be the third ventricle. And you notice there's communication. There's little channels that allow cerebrospinal fluid from the lateral ventricles to drain into the third ventricle. Those are called interventricular foramina, or singular would be interventricular foramen. Just little channels here that allow the cerebrospinal fluid again to move from lateral ventricle into this centrally located third ventricle. We don't really see that on this model. Some brain models have a little opening here showing where the interventricular foramen is allowing fluid to come into the third ventricle, but this one does not. All right, so as we zoom in closer on the diencephalon, we see that most of the diencephalon is made up of a region called the thalamus. Essentially, the side walls, all the side walls of the third ventricle is this region called the thalamus. At the very center, you see a little projection. It's actually been cut. This is the intermediate mass of the thalamus. This would actually be a stalk-like structure that connects the right and left thalamus across the third ventricle. So it's a little band of fibers, again, tract of axons, that connect the right thalamus to the left thalamus through the third ventricle. Another thing we see in this particular model in the third ventricle is this red structure right here. This is the model attempting to illustrate a choroid plexus. This is a choroid plexus. Remember, a choroid plexus is a capillary bed that secretes cerebrospinal fluid. It's not the sole source of cerebrospinal fluid, but it does account for about 30% of cerebrospinal fluid. And each ventricle has a choroid plexus. And here again, you see the cut intermediate mass. As we move down from the thalamus, we get to this sort of tapering funnel-shaped region called the hypothalamus. This is the hypothalamus. And again, this is a very important autonomic and endocrine region of the brain. On the floor of the hypothalamus, you see a lump here. There would be a second lump on the other side. Since this is a sagittal section, we're only seeing a half of the brain. These two bumps would be referred to as the mammillary bodies. The mammillary bodies. What we also see here is, again, you don't actually see it, but coming off this point right here, there would be a continuation. There would be a stalk-like structure, and some of our models have this, unfortunately not this one. A stalk-like structure called the infundibulum right here, and that would lead to a pituitary gland, which sits right here in the sella turcica on the floor of the cranium. So if this was a complete model, we'd see infundibulum and pituitary gland just in front of the mammillary bodies. Okay, so again, thalamus here, hypothalamus, this tapering region here, mammillary body, part of the hypothalamus, and here would be an infundibulum and a pituitary gland if they were there. The last part of the diencephalon is this part up here, basically posterior to the thalamus, behind the thalamus, and this is known as the epithalamus. The epithalamus. And if we zoom in on it, we see that the epithalamus, again, very tiny little region just right in here. And most of what the epithalamus is this structure right here, which is a pine cone shaped gland known as the pineal gland. And again, the pineal gland is involved in circadian and circannual rhythms and the release of melatonin. All right, so those are the structures of the diencephalon. Now, below the diencephalon, we then move down to a short region called the midbrain. This is the midbrain, this region right in here. And in this particular model, we really can't see the midbrain very well. We can see a sagittal section of it, and I see two bumps here, which are part of the midbrain, but really we have another model that's much better at illustrating the midbrain, so let me substitute this out for a moment, and we'll go to a different model. I'm going to push this one off to the side for a second and bring this one in. This essentially is a close-up of the brain stem, largely focusing on the brain stem. So just to orient you, if we pull it apart, I'm going to show you the left side now, which is a little bit different from what we just saw. Just to orient you to where we were, we'll zoom in on this a little bit. Here we once again see the diencephalon that we were just talking about. There's the thalamus with the intermediate mass. And here's the hypothalamus tapering to a point. We've got the mammillary body below. What I like about this is you can, on this particular one, see the stalk-like infundibulum leading to the pituitary gland right there. Okay, so that just gives you sort of some sense of where we just were. Now we want to focus on this region right here again, which is the midbrain. And I want to put the two halves back together to illustrate the parts of the midbrain that we want to be able to identify. If I turn it around and look at it from the front, if I look at the midbrain from the front, what I want to notice are these stalk-like structures on either side leading up into the cerebrum from the midbrain. These are known as the cerebral peduncles. The cerebral peduncles. Right there. A little stalk-like structure. It's hard to get the lighting just right. A little stalk-like structure leading up to the cerebrum on each side. Cerebral peduncles. And then on the posterior side of the midbrain, in the same level, but on the back side, we see a pineal gland. Unfortunate that it's been cut in half, but there's the pineal gland of the epithalamus right there. And just below that, on the midbrain, we see this very characteristic region of 4 bumps. This region of 4 bumps is known as the Corpora Quadrigemina. The Corpora Quadrigemina. And the Corpora Quadrigemina basically consists of 2 lumps on top, a pair on top, and a pair on the bottom. The upper pairs, known as the superior, are known as the Superior Colliculi. The 2 on the bottom are referred to as the Inferior Colliculi. So altogether, it's Corpora Quadrigemina. Superior Colliculi is the upper pair, Inferior Colliculi the lower pair, and as you can see, they sit just below the pineal gland. And again, Cerebral Peduncles make up the midbrain on the anterior side. Below that, we're going to get down into the lower parts of the brainstem. Essentially, this enlarged area here is known as the pons. This is the pons. And then below that, the slightly enlarged area is the medulla oblongata. And after this enlargement, then from there on out, it's just the spinal cord. So that's what we can see on this particular model. If we go back to the model that we began with today, we can see some of those same structures. Some of those same structures that we just talked about. I'm going to back out just a bit. And so we were just talking about the midbrain here. And if now that you have some orientation, you can see there's 2 bumps right there. These 2 bumps, that's half of the Corpora Quadrigemina, right? The superior colliculus here, inferior colliculus there. There'd be 2 more on the missing side of this brain. And as we said, just below the pineal gland that we saw in that other model. All right, we don't see the cerebral peduncles very well, but as we travel down from the midbrain, of course, we do then see that enlarged pons, that swollen region called the pons. And then just below the pons, we see the medulla oblongata, and that's going to lead on out to the spinal cord. We also have a 4th ventricle in this region. Just behind the pons is another hollow fluid-filled chamber called the 4th ventricle right there. It does have a choroid plexus like all the ventricles do. And all the cerebrospinal fluid from the other ventricles, the 2 laterals and the 3rd ventricle, will end up in the 4th ventricle. And they get there by way of this channel that runs right through the midbrain, known as the cerebral aqueduct. So this channel right here that carries fluid from the 3rd ventricle down to the 4th ventricle is the cerebral aqueduct. From here, the cerebrospinal fluid can exit the 4th ventricle by either going down into the central canal of the spinal cord or leaking out into the subarachnoid space around the brain and brainstem through a couple of apertures called the median aperture and 2 lateral apertures. We don't see those apertures on this particular model, and I don't believe we see it on this model either. So we do not have a good representation of those apertures. If we go back to our ventricle model, remember our ventricle model here, we had our 2 lateral ventricles. We had the 3rd ventricle, remember, in the center. That little hole in the middle would be the intermediate mass that passes through the 3rd ventricle. Down here, we see the cerebral aqueduct leading away from the 3rd ventricle down into the 4th ventricle, which is this structure right here. There's our 4th ventricle. All right, and again, we don't see the apertures, the little openings that are leading out of it, but they do exist. Okay, the only other structure then following along our list of terms that we haven't talked about then is, again, the 2nd largest portion of the brain, the cerebellum. It's 2nd largest by volume, but it contains about half of the neurons of the brain because it's got a lot of gray matter. And here we can see the distinction between gray matter, shown in brown for some reason, and white matter, which is sort of this branching, radiating network of axons, basically. Remember, white matter, again, is myelinated axons. Gray matter is neuron cell bodies and unmyelinated axons. There's relatively little white matter here. The cerebellum is mostly gray matter, and that's why, again, 50% of the brain's neurons are here in the cerebellum. This pattern of white matter in the cerebellum is very distinctive, radiating out like branches of a tree, and that white matter pattern is referred to as the arbor vitae, the tree of life. Okay, from here on out, the list of terms tend to focus on the meninges and flow of cerebrospinal fluid. That, of course, we don't see on this model. We do have a model that tries to show the meninges. It's admittedly not the greatest model, I have to admit, but we can see some things with it. So this is what the model looks like, and it's fairly large and cumbersome, but I'm going to hold it in my hand and hopefully you can see some things. So the focus here, of course, is not the brain itself, but the meninges. And so if you zoom in real close, what we see at the surface in gray is the dura mater. The dura mater is shown in gray. And then remember, that's attached to the skull. There is no epidural space. Then there's a very tiny subdural space below the dura mater, which really isn't shown here. And then in pink, you see a second membrane. That's the arachnoid mater. Arachnoid mater in pink. Beneath the arachnoid mater, this dark space is the subarachnoid space. And if this was a really good model, you'd see lots of little cobwebby-like extensions from the arachnoid mater down into the subarachnoid space, but we don't see that here. Then finally in red, we see the pia mater, which is very soft layer right on the surface of the brain, following every contour of the brain. That red layer is the pia mater. Those are your three meninges associated with the brain. And then, again, because this is a section, this is the gray matter that we said forms the cerebral cortex right at the surface of the cerebrum. And then the rest of the cerebrum would be white matter beneath the cerebral cortex. Essentially what happens, if we turn this over and look at it from this side and zoom out a little bit, essentially what happens here, and a couple of other things to point out and make this model nice is, remember the cerebral dura mater has two layers to it, and along the mid-sagittal line of the skull, the meningeal layer pulls away from the periosteal layer, and that creates a space, right? That runs right down the midline underneath the center of your skull. That space, remember, is a vein known as the sagittal sinus, and that contains blood, of course, and this is where we drain cerebrospinal fluid in. So it's produced in the ventricles and along the way in the subarachnoid space, but it comes from the blood. Remember, cerebrospinal fluid is a blood derivative, a plasma derivative, and it all ends up back in the blood eventually in this vein called the sagittal sinus. Notice the dura mater continues down into the longitudinal fissure as the Falx Cerebri. So this extension of the dura mater down between the two hemispheres of the brain is referred to as the Falx Cerebri. All right, and so this model, with using arrows, is just trying to show us that cerebrospinal fluid begins in the ventricles, and it's produced all along the way. It's made by choroid plexuses. It's made by the cells that line the ventricles. It flows down the cerebral aqueduct again into the fourth ventricle. Once it gets into the fourth ventricle, we have little openings. That happens to be the median aperture, for instance, that little structure right there, median aperture. And then we see some arrows indicating that the cerebrospinal fluid has made it into the subarachnoid space surrounding the cerebellum in this case, and the fluid just continues to work its way up through the subarachnoid space. We see more arrows until eventually it makes it up to this sagittal sinus that runs along the midline of the brain. And what we don't see here, unfortunately, actually we do if I turn it around and look from the front, you'll notice I just get it set up right, there are areas where the arachnoid mater pokes up into the sagittal sinus. These are known as arachnoid villi. They poke up through the bottom of the dura mater into the sagittal sinus, and these arachnoid villi are what pump cerebrospinal fluid from the subarachnoid space into the sinus, in other words, back into the bloodstream. And then this sinus eventually will drain away into the internal jugular vein and drain back to the mainstream circulation. So this model is attempting to show us some of this pattern of flow of cerebrospinal fluid, again originating in the ventricles and all along this hollow pathway, draining into the subarachnoid space from the fourth ventricle through the apertures up to the longitudinal or mid-sagittal sinus and pumped into the bloodstream by the arachnoid villi that are projecting into that sinus. So I believe at this point we have indeed covered all of the terms that are on the list of terms for this unit, the brain, and that pretty much ends our discussion of the brain for this week. Next week we're going to use this same model, and we are going to look at, I shouldn't say next week, next time, we are going to use the same model and we are going to examine the cranial nerves that emanate from the bottom of the brain. Until then, stay safe and have a great week.